Assalamu alaikum. I'm Dr. Mohammad Akram, Department of Eastern Medicine, Government College University, Faisalabad. I will discuss about the functions of intestinal juice. Since the pH of intestinal juice is alkaline, it helps in neutralization of the acid present in the chyme coming from stomach. Mucus secreted by Brunner glands and goblet cells causes lubrication of the small intestine and also buffers HCl present in the chyme. Juice contains intrinsic factors which help in the absorption of vitamin B12. IgA present in the juice protects against bacteria. Enzyme present in the intestinal juice helps in the digestion of food. Regulation of intestinal juice secretion. It is regulated by neural and hormonal mechanism. Neural control vagus nerve stimulation causes increased secretion from Brunner glands and crypts of liver cone. Administration of parasympathetic drugs, physostigmine and pilocarpine also increases secretion of intestinal juice from Brunner glands and crypts of liver cone. Cutting off sympathetic nerve supply increases parasympathetic effects, results in increased secretion of the sucrose. Succus and trachus as well as increased motor activation of the small intestine, which itself is a strong stimulus for the secretion of intestinal juice. From the duodenum and upper intestine, known as duocrinin and enterocrinin, are released into acid which cause increased secretion of intestinal juice. Other hormones which cause an increased secretion are gastric inhibitory peptide GIP and vasoactive intestinal peptide VIP. Digestion and absorption of carbohydrates. Major dietary carbohydrate are starch, sucrose, and lactose. Starch is a polysaccharide, while sucrose and lactose are disaccharides. Digestion in the mouth. Salivary alpha amylase cleaves a 1,4 linkage of starch and converts it into dextrin. Digestion in the intestine. Pancreatic enzyme. Pancreatic alpha amylase cleaves alpha 1,4 linkage of dextrin and converts it into disaccharide maltose, trisaccharide, maltotriose, and oligosaccharides containing alpha 1,4 and alpha 1,6 linkage. Enzymes of intestinal epithelial cells. Maltase alpha glucosidase cleaves alpha 1,4 linkage of maltose, releasing two glucose molecules. It also cleaves alpha 1,4 linkage of oligosaccharides and releases glucose molecule. Alpha dextrinase cleaves alpha 1,4,6 linkage of dextrin, releasing glucose molecule. Sucrase cleaves sucrose and releases one molecule of glucose and fructose. Lactase cleaves lactose and releases one molecule of glucose and galactose. Clinical notes: carbohydrate malabsorption. Isomaltase sucrase deficiency. It is deficiency of sucrase. Patient cannot digest sucrose found in Eskimos and North Americans. Treatment: avoid dietary sucrose. Lactose intolerance. It is a deficiency of lactase. Patient cannot digest lactose, thus unable to drink or eat dairy products. Lactose intolerance. Mechanism of lactase deficiency is not clear. It can be age dependent. For example, in older people, acquired or temporary patient with diarrhea, or congenital deficiency of lactase since birth. Particularly common among blacks and Asians. Symptoms: bloating, abdominal cramps, and osmotic diarrhea 2 to 4 hours after eating dairy products. Treatment: avoid milk or dairy products or add lactase pill to diet. Cellulose and indigestible polysaccharides. Cellulose is a fibrous dietary polysaccharide containing beta 1,4 glycosidic linkage between glucose residues, which cannot be digested by our humans due to the absence of its enzyme cellulase beta 1,4 endoglycosidase. Absorptions of carbohydrate. The final product of carbohydrate digestions are glucose, fructose, and galactose. These sugars are absorbed by intestinal epithelial cells, carriers moving against the concentration gradient transport all of them into the cells. A transproteins that carries sodium ions also transport glucose and it requires sodium potassium ATPs which pumps sodium ions into the blood along with glucose against the concentration gradient. Digestion and absorption of protein. Proteins are digested to amino acids, gastric enzymes, pepsin, and pancreatic enzymes, trypsin, chymotrypsin, elastase, and carboxypolypeptidase converts proteins into oligopeptides and amino acids. Enzymes of intestinal epithelial cells, aminopolypeptidase, dipeptidases, and tripeptidases converts peptide into amino acids. Amino acids are absorbed through intestinal epithelial cells and enter blood. Digestion in the stomach. Gastric enzymes. Pepsin is secreted by the chief cells of stomach and break protein large polypeptide into smaller polypeptides. Digestion in the intestine. Pancreatic secretions includes bicarbonates and proteolytic enzymes. Bicarbonate neutralize the stomach acid and raises the pH of the intestinal content. Proteolytic enzymes include endopeptidases and exopeptidases. Endopeptidases cleaves the central peptide linkage of the protein chain. Examples are trypsin, chymotrypsin, and elastase. Exopeptidase break one molecule, one amino acid at a time from the carboxyl end of the peptide, carboxypolypeptidase. Enzymes of intestinal epithelial cells, proteases. Exopeptidase cleaves one amino acid at a time from the amine end of peptide, for example, aminopolypeptidase. Dipeptidases and tripeptidases cleave dipeptides and tripeptides and release amino acids. Absorptions of amino acids. Amino acids are absorbed by intestinal epithelial cells to different types of intestine Sodium amino acids transport protein which requires sodium potassium ATPase activity transport sodium ions and also amino acids. Gamma glutamyl cycle. It is a glutathione dependent process in which amino acids are transported across the cell membrane attached to gamma glutamate and released into the cytoplasm. Digestion and absorption of lipid. Digestion. Major dietary lipids is triacylglycerol. Digestion takes place in the small intestine and requires bile salts and pancreatic enzymes. Bile salts emulsify the dietary lipids. Pancreatic lipase along with cholipase convert triacylglycerol into two monoacylglycerol and free fatty acids which are packaged into micelles. Micelles are microdroplets of fat emulsified by bile salts which also contain cholesterol and fat soluble vitamins. Absorptions. Micelles get attached to the microvilli of intestinal epithelial cells which absorb two monoacylglycerol, free fatty acids, cholesterol and fat soluble vitamins. Resynthesis of triacylglycerol. In the epithelial cells, fatty acids from micelles react with two monoacylglycerol to resynthesize triacylglycerol. Triacylglycerol is released in the lymph as a component of chylomicron, which then enters blood. Clinical notes: lipid malabsorptions. Loss of lipids includes fat soluble vitamins A, D, and E and K, and essential fatty acids in the feces is known as steatorrhea. Steatorrhea can be caused by a number of conditions related to the deficiency of bile and pancreatic enzymes or defect in the absorptive cells of the intestine. Thank you very much.